Share screen. And I need eye clicker. I'm ready for spring break. I'm with y'all. I need spring break.
All I have is one test in this class
and then spring break. There you go. You could focus on that, right?
Oh, I do have
Well, it's not too bad. That's how I feel about English too.
I'm good at it. I just need to.
I'm with you. I'm with you. Okay, on. Display on.
I just pulled it all out and left it exactly as it was.
Perfect. You are so awesome. Thank you. Okay.
So with A, B, C, D, B.
That's what's actually going to make that.
If I could have written that down, because I'll never remember that. Okay. And I'm setting something up, so give me 1 minute.
Probably a little bit of both.
Here it is. I just didn't put it on my flash drive. It's so weird. That'd be good.
I'm all the way up to the last table, but I don't know what to put for micrograms, phosphate, and I want to do the standard curve.
Y'all staying at the end of class? Okay. Come on.
That's the one thing that I'm good at. I have the standard curve,
And y'all got a computer? Yes. I know. I don't know how I did that wrong, but it's not. It's not. I also teach Chem 109.
At least it's in the right like spot.
It's yeah, it's just the wrong class. I can change that. I need to go in and change that. Let me write myself a note. I think y'all can figure it out. Y'all are pretty smart. Chapter 8 from 109 to 435. Okay, I will do that. So, oh, I didn't start class. I logged in, I didn't start class. Well, a bunch of people told me they had flooding and stuff, so we'll have some on Zoom. So I need my chat. Not a bunch, but some. So please, please, please check in on attendance. Please, please. And on the Google Drive, it does say Chem 109, Chapter 8, but it's really our stuff. It's cell signaling systems. Yeah, but it's some good content there. Don't underestimate it.
It wasn't working. It was saying that
You do not need to memorize every mechanism, Zach.
So like for the mechanisms though, like how, like you say you don't need to memorize them. So like how much you just have to understand. Like I could look at a picture and tell you what's going on in the picture.
Yeah, you have to be able to tell me what's going on in the picture. Like, do y'all want me to print? Y'all want me to post some like blank ones that don't have the descriptions, and then you could practice. Okay, so I can do that. Post. Yeah, it really is. It really is. So post blank mechanisms.
You probably would.
Okay, so how's that? You think that'll help?
That would be very helpful.
Zach, you think that'll help?
Yeah.
Zach's my gauge over there.
I don't know if anything's gonna happen. We took organic one-on-one.
Oh. Oh, but like you have to be able to look at it and say, is that general catalysis, acid-base catalysis, is that specific acid-base catalysis?
Cell is a lot of this.
Cell is a lot of this, chapter 8.
Cell is like 7 and 8.
Yeah, it's a lot of, yeah. But you have to be able to look at it and like explain what's happening in the mechanism. You don't have to memorize it. You're not gonna have to draw the mechanism.
So like.
It's gonna be provided to you. If I give you the mechanism and I say, okay, where is our intermediate? You circle the intermediate.
Okay, that's
You know, like things like that. What's that? You wanted to draw some arrows? Well, you might have a chance to draw some arrows. Okay, make sure you've checked in on attendance. If you have not checked in on attendance, please go ahead and check in. Everybody's kind of straggling in, so. But I think we'll go ahead and get started now. All right, if you are online and have a question, put it in the chat. I'm going to try and keep it up. All right, so cell signaling systems. So you'll see a lot of this in molecular biology. If you end up taking that class, you'll see some of this in genetics. You see it kind of overlaps into a lot of different biologies. But cell signaling is super, super insanely important because this is how we link the outside environment to changes within the cell, right? Because there are lots of things that go on in order to have cell communication. And so an outside event happening and creating a physiological change on the inside of the cell is how us multicellular organisms operate, right? Unicellular is a little different. You do have to have cell signaling, but not at the level that eukaryotic and multicellular organisms need. All right, so if you're going to have a cell signaling system, what are our basic requirements for that? Before we do this, what are our basic requirements? You have to have a cell, okay. You have to have a ligand, right? An extracellular ligand, right? Something floating outside of the cell. What does that ligand have to do? Bind to what? Bind to a receptor. Right? Now, sometimes that receptor is in the nucleus, but we're not talking about that. We're talking about receptors that are on the outside surface of the cell. Okay, it binds to the receptor. Then generally what happens? We're going to have some kind of a change, right? That change is going to be propagated into the cell. So you have changes in intracellular proteins. It's not always proteins, but we'll say maybe signaling molecules, that's better. Okay. And I'm going to put lots of arrows because it's not one simple step. It's a bunch of things that are going to happen. And we're going to look at bunches of examples of it. And then what happens? In order to be a cell signaling system, you have to induce some kind of a physiological change in the cell. So you need a biochemical, physiological activity change. Right? There has to be a endpoint, right? What are you trying to accomplish? You have to get there. Okay, so that's kind of our big general overview of cell signaling. So what we're going to do today is we're going to talk about generally how do signaling pathways work. Then there are 4 different categories that we're going to talk about specifically, and we'll look at specific examples. There is a lot of detail in this chapter. A lot of detail. You need to start studying. Okay. My forewarning to y'all. All right.
Do you recommend like going and reading the slides ahead? Like,
I definitely recommend reading the book ahead. That's my, this is, I guess, a day late and a dollar short. But
book pains me, like so much.
So true.
it is so dense. It is so dense. But if the first time you see it is when you read it on your own, seeing it the second time in lecture is going to help so much. So much, because you've already kind of got a little bit of a base, and then you have enough knowledge to ask questions, right? If it's the first time you see it, you might not have enough time to like digest it in your brain to like actually go, well, wait, does that make sense? You know? So it really helps if you read before you come to the lecture for the topic. But I do suggest reading one way or another. Like put yourself little gummy bears on the book, you know, on each paragraph. And as you finish the paragraph, you get to eat the gummy bear. I don't know, something. I am a mom. I bribe. I totally bribe. It's okay. All right. So what do you have to have inside of a signaling pathway? You have to have those extracellular messengers, right? Those are ligands. Now, the ligands can come from anywhere. They can come from really far away. They can come from right next door, or they can come from the exact same cell. So it's just that they're extracellular. Do they have to be a protein? No. They can be all kinds of different things, which we'll talk about, but they have to trigger an eventual cellular response, right? And so what has to happen in order to initiate that cellular response? We're going to have protein structural changes. Like we have talked about how important protein structural changes are all semester long. It's not going to change, right? You'll have one conformation when the ligand is unbound, and you'll have another conformation of the receptor when the ligand is bound, right? And so beautifully, thankfully, when biochemists were naming things, the name of the receptor usually reflects the ligand that activates it. So like there's the estrogen receptor. Guess what it binds? Estrogen. There's the insulin receptor. Guess what it binds? Insulin, you know, so on and so on. So at least they did that. They did us a favor on that, because the rest of it's gonna get crazy. So signal transduction. This is what we call once you have activated the receptor, and now everything's going on inside the cell. So these are the internal signals. This is that conversion from an external signal to an internal signal, and then we start propagating some things throughout the cell. So your book says this changes information into a chemical signal. Well, it was really a chemical signal to start with. It's just converting from a chemical signal outside the cell to a chemical signal inside the cell. So usually the end result is some kind of covalent or strong non-covalent modification to a target protein. Like one we're going to talk about later today, what happens when you go to sleep and your blood sugar gets low? We've talked about this before, right? What's the result? You're going to metabolize glycogen. So glycogen is like a really long chain of glucose, 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 right? So what do? You chop off individual glucose molecules, right? And ship that out into the bloodstream, feed the brain. So where's glycogen? In the liver. Your brain is what really needs it, right? So you need to be able to cleave off the glucose in a form that can exit the cell, get into the bloodstream, and go to the brain. Right? Okay. So that's an example of one half of a metabolic pathway. But what happens on the other half when you eat? Right? When you eat, your blood sugar goes up. Now what do you want to do? Make glycogen, so that when you go to sleep, you have glycogen for your brain, right? So that's flux through metabolic pathways. So that there are some that you're down-regulating, some that you're up-regulating, and vice versa. Ion flow through the plasma membrane is super, super important. Why is that? What's the number one example you can think of that's important? Calcium for muscle contractions, things like that. What else? Sodium, potassium for neuronal signaling, right? All of your nerves fire based on the sodium potassium pumps. Cell mobility is another one that that requires an outside cellular signal. So y'all know macrophages float around in your bloodstream, right? But then when you get an infection, like bacteria gets into your skin on a cut, well, the cells that are damaged start secreting chemicals called cytokines that basically say, ah, right? And so the macrophages inside of the bloodstream pick up that chemical signal, and they actually start out as a perfect ball, right? And then they change their cell shape so much that they stick to the walls of your blood vessels and then actually squeeze in between two cells and enter the tissue. So that's a huge change in cellular structure, right? So instead of just floating around in the bloodstream, it's got to actually, you know, the pictures of macrophages that you've seen them, right? They have all those filopodia that are like reaching out and stuff, right? So that's a huge induction of change in order to allow for cell mobility. So it's kind of an extreme example. But it also, if you have things like estrogen that come in, right? What are you eventually going to change? Expression of certain genes, right? Because that estrogen is going to be picked up by a transcription factor. And that transcription factor is going to change the amount of expression of a target gene. Now, ligands can bind to extracellular receptors and induce a signaling cascade, which then eventually has an effect on gene expression in a similar way. Okay. All right. Yes.
How do you do research? I feel like you're so smart and you know so much.
I'm going to start my research this summer.
Yes. I feel like you should be doing research.
You can enjoy that.
I do enjoy it. I really, really do enjoy it. But I really, really enjoy teaching too. So.
I'm glad. You're a good teacher.
Well, thank you. All right. So what happens once you have that ligand and that ligand binds to your receptor? That receptor becomes what's called activated, right? So the most common thing that we've talked about so far, right, is when something changes, how do we induce a change? We stick a phosphate group on it, right? So that's an example of a covalent protein modification, right? Sometimes a ligand will bind to a receptor and the receptor will autophosphorylate or dephosphorylate, and that triggers the signaling cascade. You can have that ligand bind and induce protein conformational changes. So just structural changes doesn't mean that anything was added or subtracted, just means that it has a change in its three-dimensional shape. And because we've changed that shape, now it can bind to its neighbor who's sitting next door to it in the plasma membrane, right? Their shapes now match and they can link up, right? And so we call those neighbors adapter proteins. And so they're typically non-covalent binding interactions because you want it to be reversible. So as soon as your blood sugar increases and you stop making, I want to say insulin, it's not insulin, glucagon. Once you stop making glucagon, you want to stop chopping up your glycogen stores, right? So you want to be able to turn off the signal. You don't want it to be constantly on, right? And then the other thing that can happen is that we can change our rates of our protein expression, right? If you're going to change protein expression, there are two ways that you can do that. You can do that transcriptionally. So what does that mean when we say we're going to change protein expression transcriptionally? This is our messenger RNA. And there are two ways that you can regulate messenger RNA. You can look at how quickly you make it, and you can look at how quickly it breaks down. So you can just say turnover. It kind of covers both of them. And then the same thing on the translational level. Translational level is our protein level. So the same thing's true about protein, right? You can change how quickly you make it, and you can change how quickly you degrade it. So we just say protein turnover. All right, so when we talk about these signaling pathways, we're going to kind of categorize the different parts of the pathway, because we're going to learn about all those individual G protein coupled receptors, tyrosine phosphorylation receptors. We're going to talk about all a bunch of different ones. And so before we do the individual specific ones, we're going to talk about very general characteristics that all signal transduction pathways do. All right, so we know if we have a signal transduction pathway, that we have a set of biochemical reactions, which is why we're studying it in biochemistry, right? That were initiated by that ligand, the ligand binding to the receptor. And then it has to be terminated. Well, it should be terminated. We'll put it that way. It should be terminated by some kind of measurable response that the cell is going to make. So what was our measurable response in the example earlier? Was cleavage of glucose molecules from glycogen, right? That's our output. That's our measurable cellular response. Okay, so we have first messengers. First messengers are all extracellular. The first thing that has to happen is that you have to have a ligand that's outside the cell. If you have no ligand, nothing should happen. And we'll talk about disorders where something does, but nothing should happen. So that ligand binds to our receptor. Now, the receptor is going to change its conformation. That is going to induce signaling proteins in the inside of the cell to do their thing. So those signaling molecules that are inside the cell are our second messengers. Is the receptor a second messenger? No, it is between the first and second messengers. So you have a receptor right in between the first and second messenger. So think about messengers as like, if I give you a message to go take to Miss Christie in the office, right? A messenger is going to have to go somewhere. So a ligand can go somewhere, and then something inside the cell can go somewhere, but the receptor's stuck. Okay. All right, so let's look at an example. We generally classify our signaling pathway as upstream events and downstream events, kind of like upstream and downstream in DNA, same thing. So the upstream events are, for example, our first messenger, our ligand, binding to the receptor. That induces some kind of change in our receptor protein. Our receptor protein is now active and then can work on upstream signaling proteins. Upstream signaling proteins are pretty much close to the plasma membrane, right? They're kind of neighbors. They're kind of pretty close to our receptor. Then you're going to have second messengers. Second messengers are the ones that are going to take that signal from close to the plasma membrane to wherever the target protein may be, right? So it's usually a cellular location difference, right? So we have our upstream signaling proteins. They activate the second messengers. The second messengers activate downstream signaling proteins. The downstream signaling proteins are those that actually affect our target, right? In this case, what's happening to the target protein? It's being phosphorylated, right? Maybe if we phosphorylate a transcription factor, the transcription factor is no longer active, or vice versa. It could be that we phosphorylate a transcription factor and that's what activates it. Doesn't matter. So you can have many, many, many different downstream targets, right? It doesn't have to be just one target. Sometimes we get some kind of extracellular signal and we need more than one thing to happen in the cell. Well, that absolutely can happen, right? So depending on what the signal is, will tell you what your downstream effect inside the cell is. All right. Okay. So let's kind of go down the pathway. We're going to start at the beginning. We're going to start at first messengers. So one very, very common first messenger are hormones. So hormones are typically made in one organ. They enter the bloodstream, the circulatory system, and they go all over and they come into contact with target cells in a different location and activate those receptors, right? So you typically have a gland, let's say like the adrenal gland. The adrenal gland secretes epinephrine, right? What does epinephrine do? Like this is the bear walks into the classroom in the back. Fight or flight response. So our heart rate goes up, our breathing rate goes up, our eyes dilate. We metabolize glycogen, right? All kinds of stuff is going to happen. So how many different cellular target cells did I just say? Heart tissue, lung tissue, eyes, you know, like lots of different targets. But there are some hormones that are very specific, that have certain targets. So it just depends what the ligand is, what it's going to do. But there are three kinds of categories of hormones. There are hormones that are endocrine hormones. Endocrine hormones are made in one place and travel far to their target. So maybe they're made in the brain and the target is the liver, right? Something really far away. Paracrine and autocrine, their targets are close. In paracrine, the cells are basically within the same organ. So the cell, so the manufacturing cell, is that right? FAC, that's a C. Cell is close to the target cell. Right? So in this case, here's one particular, where are they doing it? One particular, the blue cell is making this growth factor, right? This growth factor is then going to affect a different cell type, but basically in the same organ. So it's a nearby cell. That is paracrine, right? So that is this one. If you have something that's autocrine, auto meaning self, right? So one cell secretes it, secretes hormone, and the same cell type picks it up. Right? So here, this blue cell has made this growth factor and the same cell type has picked up that growth factor. So some ligands can do both, some ligands can only do one. There's all kind of craziness. Okay. All right. So just because you're a first messenger does not mean that you're a hormone. That's kind of basically what we've been talking about a lot, but not true. So we talked about adrenaline, right? And adrenaline's made in the adrenal medulla. And where does it go? They said heart cells increase your heart rate, liver cells to help you metabolize glycogen, right? But this table is only so big, so they only put a few, right? But there are others, like we talked about insulin. Where's insulin created? Pancreas. But what's its target? Well, you have glycogen in your muscle, you have glycogen in your liver, and depending on, you actually want to store some of that glucose, you're going to have an effect on adipose cells so that they can take up that glucose and convert it into fat and then store it that way, right? But you can also have ions that are your primary messengers, your first messengers. So if you're talking about neuronal signaling, right? You pump it out, it's going to target the same cell. So that's going to be autocrine, right? You can have small molecules like nitrous oxide, like carbon dioxide, right? And they can have varying effects, and we'll talk about some of those. But sometimes it's other things that we eat, like amino acids that we have in proteins, right? Those are things that we want to be able to say, I need to pick these up. I need to metabolize these. I need to build them and make more cells, whatever it is, right? So there are lots and lots and lots of different first messengers. Do you have to memorize this table? Absolutely not. We will talk about some individual examples, and those you will be responsible for, but you do not need to memorize this entire table. Okay. So here's one example of a first messenger. And our first messenger, in this case, is nitric oxide. So in the brain, you're going to have some kind of a neuronal input, an electrical firing, right? That is going to activate an enzyme. And this enzyme is nitric oxide synthase. So in nitric oxide synthase, you convert arginine to citrulline. But the big thing is that you make nitric oxide. Nitric oxide works on guanidinylate cyclase. So if it's a cyclase, it converts a nucleotide into a cyclic nucleotide. So this is GTP. So this is 3 phosphates. We're going to convert it to its cyclic form. So in that case, we lose 2 of the phosphates and we're only left with 1. So that's a monophosphate. And the little c means it's cyclic. So this is a very, very common messenger. A cyclic form of a nucleotide, very common messenger because it behaves very differently in the cyclic form than in the normal form. So this cyclic GMP will go on and affect another protein. This protein is protein kinase G. Protein kinase G is what will affect our actual target. So you don't need to know exactly what the target is, but eventually it leads to vasodilation. So this is really, really important because if we can prevent cyclic GMP from degrading, we can have persistent vasodilation. So what was done in the research is they thought, okay, what if you have patients who are having a heart attack, right? What do you want to do? The problem with a heart attack is that your arteries inside of your heart are narrow, right? Because they have clogs inside of them. So if you could dilate those blood vessels, now more blood could get through until you could get a stent placed or whatever it is, right? So they developed some drugs to inhibit degradation of cyclic GMP. Here's an example of it, right? So this is cyclic GMP phosphodiesterase. It prevents cyclic GMP from turning back into GMP. So you basically have this hang out in the cell for a long time. Well, what they found out is it didn't really work for heart attack patients at all. Anybody know what it did work for? It's a little blue pill. Erectile dysfunction. Sure did. So it was a side effect. It wasn't what they initially planned it for, but they found a huge market for it, right? So this is how Viagra was made, discovered, created, whatever it is, right? So, you know, when you think about science and you think about science research, sometimes you do research and you don't necessarily find the solution to the problem you were trying to fix, but sometimes you find some other interesting things along the way. And that's kind of how science really works, Okay, so let's talk about some other second messengers. So we have our primary messengers, right, our ligands, and then we have our receptor that changes its conformation. So then downstream from that are our second messengers. So our second messengers, their whole job is to amplify signals, right? Because maybe you have 1 or 2 ligands that are actually going to bind to a cell because you have so many cells, right? If you had to have 1,000 ligands bind every single cell, you would have to make so many ligands. But if we have something inside the cell that can amplify the signal, then you only have to have a few ligand molecules actually bind to the receptors on the cell, and then you can induce a wide scale change on the inside of the cell. So very common second messengers, like we were talking about, are cyclic GMP, cyclic AMP. Diacylglycerol is a big one, Inositol 1,4,5 triphosphate, which you'll see abbreviated as IP3. And I'm going to tell you how we generate these in a minute. And then calcium. Calcium can be a first messenger and it can also be a second messenger. It just depends on its location. Okay. So this is, if I can move this, right? This is our conversion from ATP, right? One of our secondary messengers is cyclic AMP. So what happens is you cleave off two of these phosphate groups, and then this oxygen here comes around and binds to the sugar ring, right? So it's, that's our cyclic version. And so you can have cyclic AMP, cyclic GMP, right? Those are our two big ones that we're going to see in the cell. All right. So, oh, and the other thing I think I didn't say, right, is that you can then cleave it and now your signal stops, right? That's what you want. You want to induce a really big signal. This is the signaling molecule right here, cyclic AMP, that is going to magnify the signal. But if you have something that magnifies the signal, you want to be able to turn it off really, really quickly. And so you have enzymes that will actually cleave that bond, and you can revert back to normal, regular old AMP. Okay. All right, so let's talk about an example here, right? So in this example, we're going to talk about, let's say this is glucagon. Why am I having a hard time with that today? I don't know. So let's say you have glucagon. What's the whole point of having glucagon? I want to induce what? Look at the bottom, the very bottom target. We're going to go from glycogen to glucose 1-phosphate, and our glucose 1-phosphate can go into the bloodstream, and we use it as an energy source. Okay, so that's our start and finish. But what has to happen in the meantime? Glucagon is going to bind to our receptor. We get receptor activation. We're going to have a few things happen near the plasma membrane. Once those upstream events happen, now we're going to activate a secondary messenger. Second messenger. And glucagons are what? First messenger, right? That's our ligand. So our second messenger that gets activated is adenylate cyclase. Adenylate cyclase, what do you think the cyclase tells you? You turn something into a cyclic molecule. What molecule? Adenyl, right? ATP. That's what you're going to turn into it. So if you want to know what the enzyme does, just look at the name. So we convert ATP to cyclic AMP. Yep.
So an enzyme can be a second messenger?
Yeah, absolutely. Absolutely. Most, not most, but some of them. So we have multiple second messengers, right? Because you have cyclic AMP, right? You can have all kinds of different things be second messengers. Okay, so once that cyclic AMP, once that ATP is turned into the cyclic molecule, then that cyclic AMP will then go and activate another downstream target. This is protein kinase A. Protein kinase A, PKA. You're going to hear PKA often, so you should know what PKA is. If it's a protein kinase, it's going to phosphorylate something. It phosphorylates phosphorylase kinase. Phosphorylase kinase is, guess what, a kinase. It's going to phosphorylate glycogen phosphorylase. Glycogen phosphorylase is what is going to cleave the glucose molecules off of the glycogen, and you'll be left with glucose 1-phosphate. Okay, so now I want to do the math on this because I like to do the math sometimes. If you have one glycogen, right? If you have one glycogen, I had to write this down so I get this right. One glycogen is going to activate one adenylate cyclase, right? That one adenylate cyclase goes on and activates 100 cyclic AMP molecules. Okay, those 100 cyclic AMP molecules are going to go on and activate, in this case, only 50 pKa's. What am I doing? Yeah. Oh, thank you. Thank you. I see, I told you I had a problem with that today. Don't know why. Okay, so we've got 50 pKa's. 50 pKa's are then going to go in and activate 5,000 phosphorylase kinase. Right? What do you see happening? So now we're going to release 50 million glucose molecules, glucose 1 phosphate molecules. For one ligand. That's effective. That's very, very effective, right? It works. The system works. I thought that was kind of cool. I had to write down the numbers on that. I want to get that wrong. Okay, so how does this cascade happen? Remember how we said there were some secondary messengers like diacylglycerol, DAG? Do you remember that from the previous slide? And IP3. So sometimes what you have are molecules that are embedded inside of the membrane. In this case, phosphatidylinositol 4,5-bisphosphate, PIP2. PIP2 is embedded inside the membrane. So this is embedded in membrane. When you have a ligand bind and you have activation of effector molecules, right? One of those effector molecules is this enzyme called phospholipase C. What's it going to do? It's a lipase. It's going to cleave a lipid, right? Okay, so what happens is when you cleave the two off, now they can go and do something. So in this case, now we have two downstream signaling molecules, right? So this is another way that we can amplify signals, all right? So let's look at that cascade event. So here we have a different ligand that's going to come in and activate some receptor, right? We're going to have some downstream signaling events. Now we're going to get to PLC. What did we say PLC was? Phospholipase C. Okay, so you've got phospholipase C. Phospholipase C will cleave PIP2 into two different things, DAG and IP3. Both DAG and IP3 are secondary messengers. They can go and do some similar things and some different things. So in this case, our DAG is going to go and activate a kinase. You're going to see this so often that you're going to activate a kinase or a phosphorylase or something like that. In this case, it's PKC, protein kinase C. This is biochemistry as a second language. You have to know all your abbreviations. So whenever you see it on your slide, you see an abbreviation, write it out. It'll help you get in the habit of knowing what they are. So if this is a kinase, what can a kinase do? It can phosphorylate whatever the target, downstream target proteins are. Now, IP3 is pretty interesting because IP3 can go on and activate calcium channels. Those calcium channels are in the endoplasmic reticulum. And so now we basically have calcium being a second messenger because that's now free floating. That calcium can do two things. It can actually bind to a protein called calmodulin. So it modulates things using calcium, calmodulin, kind of a cool name, right? So a lot of times calmodulin is ineffective unless it's bound to calcium. So when calmodulin is bound to calcium, then it induces some kind of effect on our target protein. right? The other thing that calcium can do is calcium can then go and activate protein kinase C, which then just synergistically has this effect on our target protein, right? So these are big general things that happen in a lot of different signaling pathways. And then we'll talk about some individual pathways in a little bit. Okay, so in the next sections, we're going to talk about the different classes of receptor proteins in eukaryotes. So the first one, we're going to talk about our G protein coupled receptors. So that means you have a receptor molecule, and that receptor molecule associates, is coupled to what's called a G protein. So when that receptor becomes activated, you're actually going to disassociate that G protein from the receptor. And that disassociation event is what then leads to activation of our adenylate cyclase, of PLC, right? Of our downstream effectors. The next class are receptor tyrosine kinases. What does this tell me about the receptor? If it's a receptor tyrosine kinase, it's going to phosphorylate, right? If it's a kinase, it phosphorylates. Well, if this is a tyrosine kinase, it's going to phosphorylate on a tyrosine residue, right? So it's going to phosphorylate some kind of target protein. And now that that protein is phosphorylated, now that phosphorylated protein can bind to something else. And we call that docking sites, right? And the third class, big class, are the tumor necrosis factor receptors. So these basically are receptors that hang out on their own and don't do anything. But when ligands bind, they come together and form trimers, so 3 receptors together, and that association between those 3 receptors together will induce downstream effects like inflammation and apoptosis, which is really, really cool stuff. Really cool stuff. All right. Then we have nuclear receptors, right? Nuclear receptors typically pick up things like hormones, right? And so they modulate our gene expression. So if the ligand binds to the transcription factor, maybe now you can transcribe that gene more, or you transcribe it less, whatever it is. But it's usually through protein-protein or protein-DNA interactions. Like usually it's not just that one transcription factor at the promoter of the gene, right? Usually it's a whole bunch of other proteins. And so maybe to associate with that collection of proteins that induces transcription, you have to have the ligand bound, something like that. And then we have ligand-gated ion channels. So if the ligand is present, then now potassium can flow, right? If the ligand's absent, the channel's closed. It's like paying the toll. The ligand is like the toll to get across the bridge, right? Okay. So here are our different classes. We have our G protein coupled receptor. So do you see this portion of the molecule? This is the receptor, this is the ligand. And this other blue thing is the G protein. And then the G protein typically has other things associated with it. So this is basically, I don't like how they quite did this, because usually once your ligand bounds, now this stuff disassociates, but they're showing it associated. But I'll let them pass. Receptor tyrosine kinases, typically when they bind to their ligand, sometimes like this one will phosphorylate this one, and this receptor will phosphorylate this one. And now that you're phosphorylated, now we have docking sites for adapter proteins. So these adapter proteins will specifically bind to phosphorylated tyrosine residues. So then that creates a downstream effect. Here's our tumor necrosis factor receptors. So remember, they usually hang out individually, but when they bind to their receptors, they trimerize, and they have interactions between their death domain portions of the molecule. And so when those death domains get together, guess what happens? You're going to activate adaptive proteins that are going to signal apoptosis. That's why they call them death domains. I thought that was kind of interesting. I feel like it'd be nice to name something, but I would probably be really boring, and I'd probably name it like really by what it does or what it. I would. I would. It's nice. But the temptation is there. I will tell you, I feel the temptation. So then you have steroids, and remember, our steroids don't have to get through a membrane, right? They can just diffuse across the cell, plasma membrane, so, and the nuclear membrane. So then they're going to bind to the nuclear receptors, and those nuclear receptors then control gene regulation, right? Okay. So how much time we got? Yeah, I'm thinking, like, I don't want to start a new one and then I'm going to get, but this is, so let's just do this. And this is the last slide. This is it. Okay. So one example of our ion channels, because remember we said that was one of our other classes. You actually see at synapses. So this is a neuron's axon terminal, right? So the very, very end. And at the very end, you have these pockets of neurotransmitters. So that's what the little triangles are. And when an electrical signal comes down and finally reaches the end of the neuron, basically what it makes those synaptic vesicles do is fuse with the plasma membrane. They release their neurotransmitters. The neurotransmitters float across the synaptic cleft, which is a tiny, 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 tiny space, and then bind to channels on the target. So in this case, we have a neuron and we have a muscle cell. And so this neurotransmitter is acetylcholine. And so when acetylcholine binds to those receptors, what happens is it opens the channel. So it's both a receptor and a channel. So when the ligand is unbound, nothing can flow through. And so your muscles aren't going to contract. But when you have a nerve signal that says, hey, I would like to raise my hand, right? You're going to merge your synaptic vesicle with the end of the neuron. It's going to bind to the channel that's also a receptor. The channel will open, and now you can allow ions to come in, and you're going to trigger muscle contraction. All right? Okay, I'm done. Because that is, yep, we'll start G protein next time. It's a really, really interesting chapter. It's just highly detailed. Y'all did? You took what? Oh, molecular cell. So how many have taken molecular cell? You're in it right now. You've taken it. Physiology. Oh yeah, this is so for some of y'all, this is, you've seen it 2 and 3 times.
I remember
Because you got it. What you got?
I forgot to check in.
It's still open. Check in.
Last class Monday.
Do me a favor and send me an email that says that, because when I'm at my desk, I can fix it because I know you were here.
Yes.
So yeah, no problem. And congratulations. Did you get?
Yeah, I got the Colonel Chem.
The Colonel Chem, that's what it was. I couldn't remember which one it was.
Yeah.
Congratulations.
Thank you.
You too. Okay, y'all want to work on lab report? You're gonna start early. Yeah, we did finish a few minutes early, but I think it's better. It's a good chunk. Look, it's good. Make it stop. It is a lot of biology. Okay, I'm gonna end this.